Hello everyone, welcome to ITLS Academy. Our today's lecture is Serology and this is part 1 in which we will discuss direct serologic testing. So let's start. First of all, we will see what are the objectives of this particular serological test. So after completing this exercise, the student will be able to perform the following objectives. Number 1. Introduction to serological testing. In this particular objective, we will define serology, antigen and state what may act as an antigen. We will also define antibody and state where they are primarily found in the body. And next step we will define direct serologic testing and indirect serologic testing. In this particular lecture we will discuss direct serologic testing and in our next lecture we will cover indirect serologic testing. Second objective is using antigen antibody reactions in the lab to identify unknown antigens such as microorganisms. In this particular objective, we will define antiserum, describe two ways of producing known antiserum, and describe the concept and general procedure for using serologic testing to identify unknown antigen, or we can say direct serologic testing. Third one is examples of serologic tests to identify unknown antigens. In this objective, we will see two parts, that is discussion and result. In discussion, we have to describe how to determine serologically whether an organism is a subgroup A, B, C or D Shigella. It will also describe how to determine serologically whether an organism is a Lancy field group, that is A, B, C, D, F or G, Streptococcus. Apart from that, we will describe how to diagnose pregnancy serologically and briefly describe the direct fluorescent antibody techniques. Next comes results. We have to correctly interpret the results of the following serological test. We will do serological typing of Shigella, serological typing of Streptococci, serological testing for pregnancy and a direct fluorescent antibody test. So let's begin. We will see introduction to serologic testing. What is serologic testing and why we perform this test? So there are two major branches of the immune response. First of all, we will see immune response branches. First is humoral and second one is cell-mediated immunity. In humoral immunity, the production of antibody molecules in response to an antigen mediated by B lymphocytes, while in cell-mediated immunity, the production of cytotoxic T lymphocytes, it is T lymphocyte, and cytokines in response to an antigen mediated by T lymphocytes. This is the major difference between humoral and cell-mediated immunity, that humoral is mediated by B while cell-mediated immunity is mediated by T lymphocytes. To understand the immune responses, we must first understand what is meant by the term antigen. So let's see what is antigen. The point is, an antigen or immunogen is a substance the body recognizes as a non-self and towards which it mounts an immune response. We can say that chemically, antigens are large molecular weight proteins including conjugated proteins such as glycoprotein, lipoprotein and nucleoproteins, and it can also be polysaccharides, and it can include lipopolysaccharide. The next point is, the body recognizes an antigen as foreign when the antigen binds to the surface of B lymphocytes and T lymphocytes by way of antigen-specific receptors having a shape which corresponds to that of the antigen. The antigen receptors on the surfaces of B lymphocytes are antibody molecules called SLG. The receptors on the surface of T lymphocytes are known as T cell receptors. You can see this is the structure of a antigen, or we can say this is a structure of polysaccharide. You can see this is these are sugar molecules. This is a polypeptide chain. This is sugar molecule, and these are epitopes. This is a structure of polysaccharide antigen. As we know that saccharide means sugar moiety, so polysaccharide antigen will contain sugar. Here this is protein antigen, and protein antigen, this will also contain epitopes, but there is no sugar moiety is present. This is a structure of antibody molecule. You can see this is heavy chain and this is light chain. This is hinge. We can say this is a region that is a connection that forms a connection between these two chains. This is Fab segment and this is FC segment. Fab segments binds any incoming antigen, or we can say it binds epitope, while the FC segment binds to phagocytes and it activates complement pathway. Antibodies or immunoglobulins are specific protein configurations produced by B lymphocytes and plasma cells in response to a specific antigen and capable of reacting with that antigen. Antibodies are produced in the lymphoid tissue and once produced are found mainly in the plasma portion of the blood. There are 5 classes of human antibodies like IgG, IgM, IgA, IgD and IgE. You can memorize this by a mnemonics that is GAMED, G-A-M-E-D. This is a descending order of size of antibody molecules. G is the largest, G, A, M, E, D. That is IgG, IgA, IgM, IgE and IgD. The simplest antibodies, that is monomers such as IgG, IgD and IgE are Y-shaped. This is the simplest shape, or you can see this particular structure shown over here is the simplest shape of the antibody. Serology refers to using antigen-antibody reactions in the laboratory for diagnostic purpose. Its name comes from the fact that serum, the liquid portion of the blood where antibodies are found, is used in testing. Our next part is using antigen-antibody reactions in the laboratory to identify unknown antigens such as microorganisms. The first point is preparation of known antisera in animals. Preparation of known antiserum in animals involves inoculating animals with specific known antigens such as a specific strain of a bacterium. Next point is preparation of known antibodies by monoclonal antibody technique. In the monoclonal antibody technique, an animal is injected with the specific antigen for the antibody desired. After appropriate time for antibody production, the animal's spleen is removed. The spleen is rich in plasma cells and each plasma cell produces only one specific type of antibody. Third point is the concept and general procedure for direct serological testing. A suspension of the unknown antigen to be identified is mixed with known antiserum for that antigen. One then looks for an antigen-antibody interaction or antigen-antibody reaction. Fourth point is detection of antigen-antibody reactions in the laboratory. Some of the commonly used techniques for observing in vitro antigen-antibody reactions are briefly described below. These techniques are agglutination, precipitation, complement fixation, enzyme immunoassay, radioactive binding techniques, and fluorescent antibody technique. Next one is examples of direct serologic tests to identify unknown antigens. This type of serologic testing uses known antiserum or antibodies to identify unknown antigens. 4 such tests will be looked at in lab. First is serological typing of Shigella. There are 4 different serological subgroups of Shigella present. Known antiserum is available for each of the 4 subgroups of Shigella and contains antibodies against the cell wall or O antigens of Shigella. Next is serological typing of Streptococci. Many of the Streptococci can be placed into serological groups called Lancy-Feld groups based on carbohydrate antigen in their cell wall. Next is serological testing to diagnose pregnancy. The hormone human chorionic gonadotropin produced by the placenta appears in the serum and urine of pregnant females. And next point is identification of microorganisms using the direct fluorescent antibody technique. Certain fluorescent dyes can be chemically attached to the known antibody molecules in antiserum. Okay, let's come to the procedure. First of all, we will see serologic typing of Shigella. First point is using a wax marker, draw 2 circles on each of 2 clean glass slides. Label the circle A, B, C, D. Next point, add 1 drop of the suspected Shigella to each circle. Third, now add 1 drop of known Shigella subgroup A antiserum to the A circle. 1 drop of known Shigella subgroup B antiserum to the B circle. 1 drop of known Shigella subgroup C antiserum to the C circle and 1 drop of known Shigella subgroup D antiserum to the D circle. This can look something like this. If these are circles, you have to put group A antiserum to the A circle while group B antiserum to the B circle. Next point, rotate the slide carefully for 30 to 60 seconds. Agglutination of the bacteria indicates a positive reaction. No agglutination is negative. Dispose of all pipettes and slides in the disinfectant container. Now come to the serologic typing of streptococci. Very first point is the cell wall antigens of the unknown streptococcus used in this test are extracted by mixing the organism with extraction enzyme. This step has been done for you. Next step, place 1 drop of the appropriate known streptococcal monoclonal antibody or latex conjugate group A, B, C, D, F and G on the corresponding 6 circles on the slide. Next point, add 1 drop of the extracted antigen from the unknown streptococcus prepared in step 1 to each circle. Next point, spread the antigen antibody mixture over the entire circle using separate applicator stakes for each circle. And fifth point is rock the slide back and forth for no longer than 1 minute and look for agglutination. Serologic testing to detect pregnancy. First point, fill the disposable pipette to the line with the urine and dispense the urine into the add urine well. Next point, shortly after the urine is added, a blue color will be seen moving across the read results window. Third point, the test results can be read in the read results window when a distinct blue line appears in the test complete window approximately 5 minutes. A plus sign indicates a positive test. A minus sign is a negative test. Next one, the direct fluorescent antibody technique. Observe the demonstration of a positive direct fluorescent antibody test. For the results, first one, result for serologic typing of Shigella. Make a drawing of your result. In these results, agglutination of bacteria is positive while no agglutination of bacteria is negative. This is Shigella typing slides. For the results of serologic typing of streptococci, make a drawing of your results. Streptococcus typing slide. For the results of serologic testing to diagnose pregnancy, make a drawing of a positive pregnancy test. You can see this is positive pregnancy test. This line is indicating positive or negative result. You will detect this test and will draw a drawing of a positive pregnancy test. Fourth one is the direct fluorescent antibody technique. For this, make a drawing and describe a positive direct fluorescent antibody test. So thank you very much guys for watching this session. This was first part of the serologic testing and we have studied direct serologic testing in this lecture. Thank you very much for watching this session. Guys, follow ITLS Academy on YouTube. You can like, share and subscribe the ITLS Academy. For any help or query, you can call us on our helpline number 7080833450. You can also reach us on www.itlsacademy.com. Thank you very much.